আচ্ছা, এমনি তোমাদের দুজনের মধ্যে কারো কোনো টপিক সম্বন্ধে কিছু ইয়ে আছে, কোয়ারি আছে? জিন এক্সপ্রেশন আর কি? হোমিওস্ট্যাসিস অফ হোমিওস্ট্যাসিস অফ হোয়াট? মেটাবলিজমে হোমিওস্ট্যাসিস হয় না। মেটাবলিজমে হোমিওস্ট্যাসিস বলে কোনো কথা হয় না। সামনে বস, লাস্ট ক্লাস তো, সামনে বস। আচ্ছা, যেটা বললো আমি সেটা আগে একটা বলে দিই, দেখ ভালো করে। ইন দি মিন টাইম যা যা ইম্পর্টেন্ট, যা যা মনে হচ্ছে ইয়ে হবে, সেগুলি জিজ্ঞেস মনের মধ্যে রাখ, জিজ্ঞেস করবি। খুব তাড়াতাড়ি আমরা সবকটা চ্যাপ্টার কভার করার চেষ্টা করবো। জিন এক্সপ্রেশন সম্বন্ধে ও জিজ্ঞেস করলো, ঠিক আছে? ডিটেলসে আমি কিছু বলবো না, কারণ ডিটেলস বলার মতন এটা ক্লাস নয়। আমি টপিকগুলো হাইলাইট করবো, হাইলাইটেড টপিক, যেগুলি থেকে কোশ্চেন আসবে বা যেগুলি কোশ্চেন আনসারের সময় তোমাদের যেগুলি অ্যাড্রেস করতে হবে। তো কি হবে জিন এক্সপ্রেশন মানে? জিন এক্সপ্রেশন মানে হচ্ছে এর দুটো পার্ট আছে, প্রোক্যারিওটিক এবং ইউক্যারিওটিক। প্রোক্যারিওটিক জিন এক্সপ্রেশন একরকম হয়, ইউক্যারিওটিক জিন এক্সপ্রেশন অন্যরকম হয়, ঠিক আছে? সাধারণত ইউক্যারিওটিক জিন এক্সপ্রেশন তোমাদের জিজ্ঞেস করা হয় না, জিজ্ঞেস করা হয় প্রোক্যারিওটিক জিন এক্সপ্রেশন। প্রোক্যারিওটিক জিন এক্সপ্রেশনের মধ্যে if not otherwise specifically mentioned that eukaryotic gene expression explain করো, অর্থাৎ তুমি সবসময় ধরে নেবে it is about prokaryotes. So prokaryotic gene expression এর আমরা কি কি দেখবো? একটা হচ্ছে রিপ্রেসার, মানে রিপ্রেশন বা রিপ্রেশন করতে পারো, রিপ্রেশন, ডিপ্রেশন। এই তিনটে মোড কি বললাম? রিপ্রেশন, ডিরিপ্রেশন, ক্যাটাবলাইট ডিঅ্যাক্টিভেশন আর or attenuation, ঠিক আছে? অ্যাটিনিউয়েশন, এই এর মধ্যে এই দুটো আমরা এক্সাম্পল দেব ল্যাক অপেরন সম্বন্ধে। আচ্ছা, একটা জিনিস, আরেকটা জিনিস আমি কোশ্চেনটা দেখলাম আনসারে, সেটা হচ্ছে যখন তোমাকে অপেরন মডেলটা এক্সপ্লেন করতে বলা হয়েছে, সামনে বস, যখন অপেরন মডেলটা এক্সপ্লেন করতে বলা হয়েছে, তখন তোমরা এই ছবিটা আঁকার সময় এই ভুলটা করছো। মনে রাখবে, যদি এটা ডিএনএ তে হয় তাহলে জিনগুলো এইভাবে লিখতে হবে, ল্যাক G, lag Y and lag A. Here লিখলাম all small. Gene is always small letter. Product capital letter. এখান থেকে যে প্রোডাক্ট তৈরি হবে সেই প্রোডাক্টটা হবে এখান থেকে যেটা হবে পারমিয়েজ, সেই প্রোডাক্টটা বা আরএনএ নাম হবে, ঠিক আছে? এখান থেকে যে প্রোডাক্টটা হবে সেটা হবে এই, যখন ডিএনএ মধ্যে লিখবে স্মল লিখবে, খেয়াল রাখবে। এটা কিন্তু অনেকে ভুল করেছে, যেই বইতে তোমার পড়েছো সেই বইটা তাদের ভুল দেওয়া আছে। ছোটখাটো ব্যাপার, এর জন্য তোমার কেউ ফেল করবে না, ঠিকই আমরা নম্বর দিয়ে দেবো, কিন্তু সঠিকটা জানা উচিত। ইউ মাস্ট নো দ্য রাইট থিং। তাহলে দুটো আছে, রিপ্রেশন ডিরিপ্রেশন, ক্যাটাগরি জেনারেটিভেশন আর অ্যাটেনিউয়েশন। এই দুটোর এক্সাম্পল হচ্ছে ল্যাক অপেরন, আর এটা হচ্ছে ট্রিপ্টোফ্যান অপেরন। মনে থাকবে? তাহলে এখানে আমরা দুটো পয়েন্ট বলবো। এক হচ্ছে রিপ্রেসার মলিকুল তৈরি হচ্ছে কোথায়? পিআই, ল্যাকাই, এই প্রোমোটার অপারেটর রিজিয়ন। প্রোমোটারের পরে লাইনটা স্ল্যান্ডিং হবে, ছোট, পিও, প্রোমোটার আর অপারেটর রিজিয়ন। অনেকে এটাকে লম্বা করে তা নয়, রং। ওভারল্যাপিং রিজিয়ন অফ প্রোমোটার অপারেটর রিজিয়ন, এখান থেকে যেটা তৈরি হবে সেটা হচ্ছে আর বা রিপ্রেসার। এই রিপ্রেসার সাধারণত ডাইমার হিসেবে তৈরি হয়ে থাকে। ইট ইজ অ্যাক্টিভ when it is tetrameric. Tetrameric form binds here and thereby it physically hinders the binding of sigma factor. So RNA polymerase cannot sit and cannot initiate transcription of the gene. So no more polycystinic mRNA will be produced. Clear? Now how do you defunct this repressor molecule, inhibit repressor molecule? You need inducers. Inducers কি কি আছে? ন্যাচারাল এন্ড আর্টিফিশিয়াল, দুরকম। ন্যাচারাল কি? অ্যারোল্যাকটোজ। আর্টিফিশিয়াল কি? IPTG, isopropyl thiogalactoside, isopropyl thiogalactoside. So either of these, অ্যারোল্যাকটোজ or IPTG. যদি IPTG হয় তাহলে IPTG চারটে মলিকুল লাগবে। 4 molecules of IPTG will bind like this. Okay, and they will dissociate repressor molecule from tetrameric to dimeric form, and hence they cannot bind promoter operator region. So transcription will proceed. এই সামনে বস, আর যদি এমনি বসতে হয় তাহলে সই করে দেওয়া হয়েছে বাড়ি যা। Last class. Okay, so 2 points you have to mention: repression by tetrameric repressor molecule, depression by presence of inducer. Two types of inducer, natural and artificial. Natural কি আছে? অ্যারোল্যাকটোজ, artificial IPTG. Next হচ্ছে category gene activation product. CAP, CAP-CAM complex, formation of CAP-CAM complex, ठीक है, which inhibits ল্যাকাই, মানে রিপ্রেসার মলিকুল তৈরি হয় না। ঠিক আছে, 1 নম্বর। 2 নম্বর হচ্ছে দুটো ভাবে কাজ করে। এটা কি করে? এটা এই জায়গাটায় গিয়ে বসে। যে ক্যাপ ক্যাম কমপ্লেক্সটাকে আমি একটু কালার দিই। এটা এইভাবে বসে। বসলে কি হবে? The repressor molecule cannot occupy the same place. Repressor molecule 3টে রিজিয়ন থাকে, বাদ দাও। OR1, OR2, OR3, ঠিক আছে? ওইখানে যদি 1 আর 2 এর মাঝখানে বসে তাহলে রিপ্লেসার মলিকুল বসতে পারে না। রিপ্লেসার মলিকুল যখন ফিজিক্যালি বসতে পারে তাহলে কি হবে? জিন এক্সপ্রেশন উইল কন্টিনিউ, সিনথেসিস অফ পলিসিস্টেমিক mRNA উইল কন্টিনিউ। হলো। নেক্সট আসবে অ্যাটেনিউয়েশন। অ্যাটেনিউয়েশনের জন্য আমাদের ট্রিপ্টোফ্যান অপারেন্টের মডেলটা এক্সপ্লেইন করতে হবে। উই নিড টু এক্সপ্লেইন tryptophan operon model. Remember, just because we are not mentioning repressor molecule, repression and de-repression or catabolic gene activation in case of tryptophan operon, that does not mean that tryptophan operon has no repressor molecule. It does have a repressor molecule, but it is more conventional to explain the repression and de-repression model through lac operon than tryptophan operon, because tryptophan operon is unique for its attenuation model. ঠিক আছে? তো কি হয় tryptophan operonের সময়? attenuation মানে কি? attenuation মানে হচ্ছে mRNA সিনথেসিসটাকে আমরা ইনহিবিট করব। কি হয়? DNA, RNA pol, mRNA is being synthesized. So mRNA is synthesized from 5' to 3' direction. At the end of the 3' direction, যদি কত রিজিয়ন থাকে, এটাকে বলা হয় পলিসিস্টেমিক mRNA সিনথেসিসের আগে একটা ছোট্ট অংশ থাকে, তাকে বলা হয় লিডার mRNA। এই লিডার mRNA তে 4টে রিজিয়ন থাকে, 1, 2, 3, 4, ঠিক আছে? এই 4টে রিজিয়নের সিনথেসিসের উপরে নির্ভর করে অ্যাটেনিউয়েশন হয়। ফার্দার সিনথেসিস অফ mRNA will continue or not will be decided by transcription of these 4 region. মজার ব্যাপার হচ্ছে in prokaryotic model transcription and translation takes place simultaneously. There is no gap in space and time in translation and transcription in case of prokaryotic model. So what happens? The moment mRNA is synthesized, the ribosomes occupy and the protein synthesis begins. Fine? So at the end of this region, there are multiple tryptophan 1 and 2. One region-এর শেষের দিকে multiple tryptophan residue থাকে, কোডন। If there is less tryptophan in the system, in the medium, then what will happen? The ribosome, there will be less number of tryptophanol tRNA. So ribosome will halt, will stop and wait for tryptophanol tRNA to come. Why? Because tryptophan is less. And this operon is responsible for biosynthesis of tryptophan. So ribosome will stop and wait for tryptophanol tRNA to come. So what will happen? The RNA pol will continue the synthesis and ribosome will stop here. But mRNA will continue synthesis 2 and 3. Now the segment 2 is unique. It has got dual property. It can base pair with segment 1 as well as segment 3. Now if it base pair with segment 1 like a loop, then it will stop RNA synthesis. Fine? If it base pairs with segment 3, then the transcription will continue. Fine? Now when 2 will bind with 3 or 1, it will be decided by which portion is synthesized and free. So if the ribosome occupies the end part of the fragment A or portion A, portion 1, then 1 is already occupied by ribosome. It cannot interact with segment 2. So segment 2 is free to interact with segment segment 3, which is being produced soon after that. Correct? Because RNA synthesis will continue. Ribosome is stopped. So 2 and 3 will join, and hence 2 will be unavailable for base pairing. It is not that 2 and 3 will base pair, that's why it continues. No. It is like that 2 is unavailable for binding with segment 1. That is why it cannot stop the transcription. Am I clear? The segment 2, if it binds with segment 1, it will stop transcription. Since it is not free to bind with segment 1, that's why it cannot stop transcription. So it binds with 3, that means it is not free anymore to bind with segment 1. So transcription will continue. Fine? This model is called attenuation. Now, in a condition where tryptophan is more in the medium, the ribosome will not wait. It will quickly synthesize segment 1 and it will go to segment 2. And before the segment 3 is synthesized, segment 2 will be synthesized of mRNA and it will base pair with segment 1, and that will send a message of termination of transcription. How? There are two ways of termination transcription. One is row dependent, another is row independent. So what is the row independent method? Hairpin loop. So 1 and 2 will form that required hairpin loop which will terminate transcription process. Clear? So these are the two models of prokaryotic gene expression. Eukaryotic gene expression is something different and much more complicated. There are few jotted points in your books. I would rather recommend you to remember the headlines, that's all. Okay? That's all. Next, anyone is having any doubt regarding any portion, any question of whole syllabus? Yes? Anyone? So now we'll discuss the important questions from different chapters. Which chapter you want me to begin with? Glucose metabolism already had been taken by HOD sir. I don't know. Glucose metabolism I will not discuss. Carbohydrate, except carbohydrate and protein. Huh? Heme metabolism, fine. Bolo. হ্যাঁ বলো হিম কি পড়াতে হবে ইম্পর্টেন্ট কোশ্চেন পড়ানো তো সম্ভব না 1 ঘন্টায় বলবো ফাইন হিম থেকে লেখো হিম থেকে ফার্স্ট যা তোমরা পড়বে স্ট্রাকচার অফ হিম ভেরি ভেরি ইম্পর্টেন্ট স্ট্রাকচার অফ হিম কে পারবে স্ট্রাকচার অফ হিম আঁকতে হাত তোলো স্ট্রাকচার অফ হিম আঁকতে কে পারবে raise your hand structure of cholesterol পারবি আমি আমার রাখি, এখন সময় নেই। Okay? You should be able to draw the structure of cholesterol with numbering of the molecules. Any doubt? You should be able to draw the structure of heme at any moment during the examination. You should be able to draw the structure any time, any moment during the examination. If you don't know this, you are not prepared for examination. There should be some parameters by which you can yourself understand whether you are ready for the exam or not. You should be able to tell the name of the essential amino acids and fatty acids. Name. Why not amino acid? Why not amino acid, essential amino acids? Quickly, don't waste time. You know it? Make it loud. Stand up. Stand up. I can't hear. No, sir. Yes, sir. Yes, you will. Last bench. Name essential amino acids. How many are there? 20. Fine. Sit down. You need not to try any further. Follow. How many? First you check. Essential amino acid. How many? Yes. 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 Next. Bolo. Whatever you want to tell it loudly. 9. Why 9? Why do you like 9 number so much that you cannot change even after hearing that it is incorrect? I told her 9 is incorrect. Still you are going on with 9. Why? If you continue with wrong answer, will it become right? Then why you don't use your brain? Next. Which number comes before 9? Thank you. So this is a parameter by which you should understand whether you should pass or not. You are not able to even utter the number of essential amino acids which you have studied probably from class 8. Next question. Name the essential fatty acids. Last bench. Number? No idea. Okay, partially accepted. Why? Because some books, they still continue to write arachidonic acid as essential fatty acid, but it is completely not essential. What is the formula of arachidonic acid? You must remember the formula of linoleic acid, linolenic acid and arachidonic acid. Always pronounce the way I am pronouncing: linoleic, linolenic, arachidonic. Linoleic is 18, 2, 9, 12. Octadeca-dienoic acid, all cis. Linolenic acid 18, 2, delta 18, 3, delta 9, 12, 9, 12, 15. Octadeca-trienoic, all cis fatty acid. Arachidonic acid, all cis, delta 5, 8, 11, 14. Docosa tetraenoic acid. Correct? This is how you must remember. Why? Because the next comes DHA. DHA is important for brain development. Docosa hexaenoic acid. 22, 6 delta starts from 4. Correct? So 4 double bond containing acids are called arachidonic acid. So another double bond system is possible. So there could be another form of arachidonic acid. That's why arachidonic acid may not be taken completely as essential, because some part of arachidonic acid can be synthesized from linoleic acid. And this pathway is also asked. Explain, like this question you will get, name the essential fatty acids. Similarly, you can also get, explain why arachidonic acid is not completely essential. Paniche. Why arachidonic acid is completely not essential? Why? This question also will be asked. Fine? Now coming to heme, you should be able to draw the structure of heme. Can anyone tell me what this MPP means? Ball. MV মানে কি? Anyone? Methyl, vinyl, methyl, propyl. This is methyl group. This is vinyl. This is not propyl, propionate. Clear? Very poor. A lot of things to study. Sit down. Now what you will study from the heme? The next thing you must remember, the heme containing proteins and enzymes. You should be able to name the proteins and enzymes separately which contain heme. Name one enzyme which contain heme. Protein you know. Name 5 proteins which contain heme. Okay, name two proteins which contain heme. Now is okay? Hemoglobin. One more. From class 10 knowledge I am asking. One more. So after studying 1 year here, you have receded 2 years backward. Huh? This is how you will progress. Myoglobin. Two you could not name. Very poor. Bujo. Enzymes. বল আগে দাঁড়া এইগুলো ভদ্র আছে সুপার ডিসমিস ফাইন কারাটি করা সর্বঘরে কারাটি করা সব জায়গায় আছে বলে মানলাম আর দ্বিতীয়টা বুঝে বলবি না বুঝে বলবি না প্রোটিন হয়ে গেল বুঝ একটা জানিস ক্যাটালাইজ জানিস না পারঅক্সিডেজ জানিস না মাইলোপারঅক্সিডেজ জানিস না জ্যান্থিন অক্সিডেজ জানিস না না জানতে হবে নেক্সট it comes heme synthesis heme biosynthesis pathway degradation pathway porphyria, different types of porphyria, classification of porphyria, LECO, practice writing at least before examination. Okay, so heme you cannot leave anything. Synthesis, porphyria, types of porphyria. How do you classify porphyria? Last bench. You don't remember. At least you could say, I don't know. Did you ever study it? If I give you the book, can you find it out? Why do you lie to yourself? Don't lie to me. Nothing will happen. Don't lie to yourself, Baba. Don't lie to yourself. You lie to everyone else in this world. Don't lie to yourself. I'm not scolding. I'm just showing you the mirror. That's all. I just want you to have good eyes. That's all. It will help you, not me. This is for you, not for me. So, say, submit. I don't know. That will hit you, that will make you study, that will make you find the answer. The moment you say, I forgot, that means you are hiding something, that your inability. Don't hide your inability with you. Make it prominent so that you can get over it. Fine, sit down. Well, types of porphyria. How to classify porphyria? Hepatic and hematopoietic. Hepatic and hematopoietic. One is mixed. Which one is mixed? Variegate porphyria. Right? Initial porphyrias are having neuropsychiatric symptoms. As you go below, from brain to blood. Right? So like this you should remember the classification of porphyria. Next comes metabolism of heme. The whole pathway you must remember. Formation of biliverdin, formation of bilirubin, transport of bilirubin to liver, entry of bilirubin into liver, conjugation with glucuronic acid inside the liver, excretion of conjugated bilirubin from the liver, pouring it out in the extracellular tissue. How? Through bile duct. Then reabsorption. Then part of it will reach the large intestine and will get excreted through stool, which will undergo oxidation by aerial oxygen and will give dark color, which is otherwise known as starcobilin. So this whole root of bilirubin you should remember. Then comes the clinical orientation. The clinical part comes with jaundice. What is jaundice? Classify jaundice. Okay? Compare the clinical findings in different types of jaundice. That's all. If you read this part, a portion of first year, for first paper, and portion of will be done. Because the chapter is in paper 1, but it can also be asked, because it has porphyria, it has jaundice. It can also be asked in paper 2. There is no hard and fast rule, like the demarcation in paper 1 and paper 2. Okay? So don't remain under such misconception that this part is paper 1. No, that is wrong, gone. Those golden days are gone, now you are grown up. Now you are grown up. Are you? You enjoy everything like adult, so enjoy the study also. Don't expect the world to behave like 2 years back with you. Since you are adult, enjoy the way the world behave with an adult. No pain, no gain. Done? The important topics of important question from him. Over. Anything else? Now let me discuss quickly with the minerals and vitamins. A very large portion of your syllabus contains this. And you will receive many questions from iron, from copper, from zinc, from iodine. Starting with minerals. You should be able to distinguish between macro and trace elements. Macro and trace elements. What do you mean by trace element? So what do you think? Iron is a trace element? একটা মাপ আছে, কিরকম মাপ শুনি একবার? কি ট্রেস না ম্যাক্রো? শিওর আয়রন ফর্ম আছে? Anyone else? ভুল বলেছে, is wrong. Very well. কি বল? কেন? সে কে? What do you mean by trace elements? No idea. Anyone? বল। কেন? চেষ্টা কর। ও চেষ্টা করলো, পারলো না। চেষ্টা কর। একটা কথা বলি শোনো, যে মাস্ক ফেসিস হয়ে কোনো লাভ নেই। পরীক্ষায় যারা ভাববে মাস্ক ফেসিস হয়ে বেরিয়ে যাব, ইউ মাস্ট ইন্টারাক্ট, রাইট অর রং, ইউ মাস্ট ওপেন ইউর মাউথ। If you don't open your mouth, that means you know nothing. If you say wrong also, that means you know wrong. If you don't open your mouth, that means you know nothing. If you open your mouth and the answer is incorrect, then that means you know wrong answer. But you know something. Maybe wrong, but you know something. But you do not open, you know nothing. So you cannot be considered. Am I clear? So next time whenever I will ask any question, try to answer right or wrong. Come on. Answer the wrong answer. It is not present in our body in little amount. It is the requirement, daily requirement is below 100 milligram. আছে লেখাপড়া করলে ভালো বলতিস ক্লাস 10 এ ট্রেস এলিমেন্ট কাকে বলে লিখলি তো না লিখলি কি বললাম জানিস না বলে দিলাম লিখবি না আবার এই ভুলটা একই ভুলটা পরীক্ষার সময় বলবি which is required recommended daily allowance the elements whose RDA is less than 100 what 100 what? Milligram. Without any no meaning. Whose RDA is less than 100 milligram. They are called trace elements. It is not the amount in present in our body. Define what are macro elements. Very good. Good improvisation. Can you name some? Fine. You know the wrong answer, but you know something. Anyone else? Anyone else? Still those who are not raising your hand, even with a wrong answer, they are missing something. They are missing the habit of answering during the examination. I am telling you. You will face very harsh consequences in the examination. The external examiners won't entertain anything. Anyone else? In মানে এনে সে কি বল phosphorus sodium না নাইট্রোজেন মেটাল নয় সোডিয়াম বোস fine now from here the first question you can expect from sodium potassium chloride although this is asked mostly in the subject of physiology. But if at all it is asked from biochemistry, then the questions will be from sodium potassium chloride. Number 1, causes of hypernatremia, causes of hyponatremia, causes of hyperkalemia, causes of hypokalemia, causes of hyperchloremia, causes of hypochloremia. Follow? 6. Hypo. Causes. Just causes you remember. Next what? What these three are called what? Electrolytes. Right? Serum electrolytes. So what are their functions? What are their functions? বলো Am I right? Am I? ঠিক আছে তো action potential, recipient potential, voltage channel, acid base balance. Now if you know all these function, how sodium, potassium and chloride are maintained, then you will find very easy. These three together, they maintain all these 6 functions. Fine? So explain accordingly. Suppose the question comes with potassium, explain accordingly. The question comes with chloride, explain accordingly. Okay? Slight difference is there, how they maintain it. Because among them, 2 are cations and 1 is anion. So their movement will be opposite. Chalk, देखो chalk आते हैं ना? Chalk. Next. Among the macroelements, not all are so important, magnesium and all. Let's come to trace elements. The first comes iron. From iron, what you are going to read? You read everything. Read everything from iron. Don't leave anything. There's storage, there are RDA, causes of low iron, loss of iron from body, right? Then supplement, if you go for supplement, then what adverse effects you can have. Then different types of anemia, this part again for physiology. Then comes absorption, factors affecting absorption of iron. Everything you have to study. Very, very important. At least 2 questions you will get from iron. Next, absorption, factors affecting absorption, then transport, then storage, then one-way metabolism. One-way metabolism of iron. What does it mean by one-way metabolism? That means iron cannot be lost from our body normally unless we have bleeding. It can enter, but it cannot exit from our body. The only other way normally is what? Exfoliation of the inner gut. Shedding of the gut mucosal cells, along with excess iron will be lost. Storage protein of iron. There are 2 types of storage proteins of iron. One is ferritin, another is hemosiderin. Among them, remember, ferritin is a dynamic store. Ferritin is a dynamic store for iron. That means accessible. From where iron can be withdrawn, from where iron can be put, stored. Unlike hemosiderin. Hemosiderin is something, ferritin, if you compare ferritin with a football, hemosiderin is a busted football in an endeavor to inflate it. You go on putting air inside the football. What will happen? It will inflate, and then in one moment the weaker portion of the sieving areas they will burst. The shape of the football will remain same, but a portion will be torn from the cover. So that is hemosiderin. And the intact football is ferritin. What is the difference? You can play with football and you cannot play with busted football. That means ferritin is dynamic. You can utilize the iron stored in the ferritin, but you cannot utilize the iron stored in hemosiderin. That is why there is a disease called hemosiderosis, not ferritinosis. That is why there is a disease called hemosiderosis. Hemosiderosis. Correct? What is hemosiderosis? What are their symptoms? What is the treatment? What are the iron chelating agents? These will be the questions asked. Fine. Now move on to copper. Move on to copper. From copper you need to remember the enzymes which contain copper and two diseases. The oxidase. মনে বুঝি না দিয়ে কি ছিল একটু ভুলে গেছি এখন মনে পড়ছে না হাইড্রোলেজ যাও at least remember these names copper containing proteins or enzymes celluloplasmin uricase lysyl oxidase tyrosinase monoamino oxidase superoxide dismutase catalase. Fine. So remember like this. Next, two diseases. Wilson's disease and Menkes disease. This is due to defect in ATPase7A and this is due to defect in 7A and 7B both. One is copper excess, another is copper deficiency. Right? Okay. Now next, read short questions from zinc, iodine, selenium, আর একটা কি আছে fluorine. From fluorine read two things, fluorosis and genu valgum. Fluorosis and genu valgum. Okay, these two. remotely sometimes they might ask about arsenic poisoning. Okay. But less important. Rather, lead poisoning is more important. Lead. Effect of lead poisoning. From vitamins, expect long question. Expect long questions means read everything. Vitamin A, D, folate. Beside folate you write 1 carbon metabolism. 1 carbon metabolism. Next. For short questions, read the functions of vitamin C, function of vitamin E, function of vitamin B12, absorption of vitamin B12, types of vitamin B12, disorders like deficiency manifestation of vitamin B6, niacin, use of biotin, riboflavin, use of biotin and riboflavin. These are for short questions. Done. From protein energy malnutrition, PEM, only 2 things you have to remember. The difference between kwashiorkor and marasmus. That's all. You read these 2 columns, sufficient. Okay. If you can read a little more, then the biological value, those things you can read, those definitions. Fine. From molecular biology, mutation definition, types of mutation, different types of repair mechanism, full replication, transcription, translation, full. You will get 2 out of these 4 topics. 2 you will get. Fine. Next. Use of RDT. There are uses in medicine or in molecular biology. Then techniques, PCR, Southern blot, DNA fingerprinting, and if little harder question comes, then sometimes they might ask about microarray. Short note, 3 marks. Microarray. Fine. Then questions might ask like this. Mention the vitamins which prevent anemia. Like this. Different way. So you should be able to answer those vitamins and their function. from lipid read classification of lipids very carefully. Very, very important. Classification of lipids. Then comes different types of fatty acids, classification of fatty acids. Then comes digestion and absorption of fat or lipid. Lipoprotein metabolism and disorders. Everything you have to study. Cholesterol biosynthesis up to metabolic. Cholesterol biosynthesis up to cholesterol. Specialized products formed from cholesterol. Specialized products formed from cholesterol. Hello? Here synthesis of vitamin D also will come. Synthesis of primary and secondary bile acids will come. Okay? They alone can be asked separately as short questions. Primary bile acids, secondary bile acids, their function, their role. Then one short question came few years back. Bile acid sequestering agent. Bile acid sequestering agent. You jete paris. You can go out. Class is over. Huh? Javi? Get down. Get down. So, bile acid sequestering agents. Fine? Next. Different types of hyperlipoproteinemia. Different types of hyperlipoproteinemia. Where chylomicron is elevated, what are their effects? Pamarjanthoma, all these things you must explain. You should be able to know these terms. When cholesterol excess, hypertriglyceridemia, hyper LDL, hyper HDL, what will happen? No HDL, zero HDL, very low HDL. Familial familial abetalipoproteinemia. Person, family-wise, familial, genetic, no one is having HDL in the family. Genetically. Like this, you want to remember. Right? In the examination, we have asked you the structure of immunoglobulin. Please draw this diagram. Always draw these diagrams. Always draw these diagrams. If you are drawing IgG, then extend it one domain further. Fine? Am I clear? This is a standard way to depict the secondary structure. It is not transcription. You should be able to draw the secondary structure of tRNA. এটা কি বলে ক্লোভার লিফ স্ট্রাকচার অফ টিআরএনএ। আমি কতগুলো ফিচার্স এখানে মেনশন করার চেষ্টা করি যেগুলো তোমার ছবি থাকা উচিত। ইউর ডায়াগ্রাম মাস্ট possess those things. What? 3' end. Okay. Each one is having 7 unpaired bases. 7, 7, 7. 7, 7, 4, 5, 7, 4, 4, 5. Base pairs. This is anticodon arm. Anticodon have a 7. This is what? এটা কি বলা হয়? Anticodon loop, DHU loop, decisive loop. Now compare if your diagram had these points, you would have got good marks. Had your diagram missing any one of those, you would have lost few marks. Fine? Clear? That's all. End of the session. There will be no biochemistry class on 12th, Monday morning. Enzyme, definition, properties, classification. Definition and classification board. Factors affecting enzyme activity. কি কি পড়বে এর মধ্যে? Under this factors affecting enzyme activity, enzyme concentration, substrate concentration, enzyme concentration, temperature, pH, substrate concentration, presence of inhibitors. Last, coenzymes, list of coenzymes, battery of vitamins and their corresponding coenzymes. This is a ideal answer. Next. The most difficult part in the enzyme is if they ask you from the units of enzyme activity and kinetics. So for those who can read little more, who can remember little more, they go for this part, steady state kinetics. Steady state kinetics. this is a graph which shows steady state kinetics of enzyme. This is substrate, initial substrate is high, then with the time it will go down below 0. And product initially it will be 0, it will increase. This is ES complex. Enzyme substrate complex. This will be 0, then slowly it will increase. Then it will maintain a constant speed. Then when the product will become this, এরকম করলে ভালো হয়। The point where it will become flat, this will increase or decrease. Enzyme সাথে হলে decrease করবে, 0 তে আসবে। আর this is free enzyme. Okay. Now this portion between these two, this is called steady state. There is no change in the concentration of ES, means DES dt 0, no change. It's called steady state kinetics. That's all. If you can draw this diagram, you will get marks. Then comes clinical enzymology. Clinical enzymology. Organ-wise enzymes you must remember. Liver enzymes, cardiac enzymes, prostate-specific enzymes. Then what? Acid phosphatase, alkaline phosphatase individually, and their different types, different isoforms, isozymes. LDH 5 types, LD 1 2 3 4 5 কি থাকে? কোনটা কোথায়? H4, H3M, H2M2, HN3, MM4. Right? H1 LD1 heart, LD5 muscle, liver also. Fine? CK creatine kinase. Creatine biosynthesis. Creatine and creatinine, don't confuse. Creatine biosynthesis require 3 amino acids, 3 organs. Then the catabolism of creatine forms creatinine. So measure of creatinine in our body is a direct measure of amount of muscle. Fine? Measure urea in our body. It's not related to the muscle. The amount of creatine in the body, urine or blood, this is related to muscle, not urea. Clear? So, the more emaciated the person is, less amount of creatine will be there in the blood normally. If not, the kidney is not functioning well properly. Right? Now, creatine creatine phosphokinase or creatine kinase, same. It's got 3 isoforms, CK1, CK2, CK3, BB, MB, and MM. Fine. CK1 will increase in meningitis. Correct? Any trauma of CNS. CK3 will increase in muscular dystrophy, Duchenne's muscular dystrophy, or surgery, or slashing injury of large muscle. Okay? CK2, cardio-specific. Fine. Now among CK2 there are two other sub isoform. CKMB2 is a sub isoform of CKMB which is most specific for heart attack. কি বললাম? CKMB cardio specific. CKMB মধ্যে আবার CKMB1 CKMB2 আছে দুটো। CKMB2 টাকে আমরা মাপি অ্যাকচুয়ালি, CKMB কে মাপি না ল্যাবে। ঠিক আছে? ক্লিয়ার? Alkaline phosphatase, different isoforms of alkaline phosphatase. Heat labile, heat resistance. Heat labile, heat resistant. হলো? আর কি? প্লাসেন্টাইল। প্লাসেন্টা থেকে এগুলো তৈরি হচ্ছে। রেগান আইসোফর্ম। এই পয়েন্টগুলো মনে রাখবেন। দ্যাটস অল ফর এনজাইম। আরেকটা কোশ্চেন আছে প্রায়শই জিজ্ঞেস করে, সেটা হচ্ছে ইউজেজ অফ এনজাইমস ইন মেডিসিন। নাম্বার 1 লিখবে ইনডাইজেশন, বিয়েবাজি খাওয়ার পরে খাওয়ার জন্য লাগে। নাম্বার 2 ক্লট লাইসিস, ক্লট লাইসিস, তাই না? নাম্বার 3 টিস্যু ইনফ্লামেশনে ব্যবহার করা হয়। ঠিক আছে? নাম্বার 4 উন্ড হিলিং। ঠিক আছে? উন্ড হিলিং, উন্ড হিলিং। ঠিক আছে? এই ব্যবহারগুলো বইতে লেখা আছে, স্ট্যান্ডার্ড বইতে লেখা আছে।